mushrooms for a little while. My name is John Carnes, and I think what I'll do is I have a bunch of things to kind of show and tell, but I'll talk first a little bit about just some general ideas. And I guess the first thing is that, I don't know if you can, radical mycology. So that would be the for me, the term for you to remember if you want to start. There's just a ton of information now on the internet. I started growing mushrooms, I think, 1978 or something like that, and I've been doing it off and on since. Many different varieties, but if you and I just got excited last year when I ran across this group of people, the guy that wrote this book, and he's from Oregon, and the radical mycology, I guess you could call it a movement, is a very hospitable, open source kind of group of people around the country that are interested in cultivation of mushrooms for a variety of reasons. But if you Google radical mycology, you'll find their website, and you can go from there. And I think I'm not on their website right now, but probably able to get a hold of me through that. Also, I started a page on Facebook, local, for like a local group, and the name of that page is Team Fungi, 2 words, and the username is Columbus Fungus. So Columbus Fungus on Facebook. Anyway, so and you could message me there, and we can stay in touch. I mean, my hope is that we'll get people that get interested. It's just it's a good way. It's a good to share some resources and equipment and whatnot. And this book is just a wealth of information. So that it's a very complete and complex discussion of the techniques. So anyway, I'll stay up here on stage for a little while, talk, and then I'll just come down there and we can pass some of this stuff around, and I'll show you what's new. It used to be a lot more difficult to grow mushrooms because when the whole thing with mushrooms are that their main function is decomposing things, right? So they're the great recyclers of the earth. And mushrooms, fungus, I should say, is a little more general term. They're responsible really for oil in the world is really critical and is largely maintained and built by fungus in one way or another. And the fungal world, the thing that's exciting to me is that it's a wide open area for amateur work. And even the word amateur comes from the Latin root of love, amo, amas, amat. So an amateur is a lover of fungus, and a lover of working with fungus. So that's really all that's required, that you're interested. And they estimate that, or it's estimated there's a 1.5 million species of fungus, of which only about 5% have been named. So there's a lot of work that can be done. And even if you go into the academic world to study fungus, in most places, they only study pathogens, primarily plant pathogens and human pathogens, things that cause problems in agriculture. But there's very little funding and work professionally even. So there's a lot that can be done, and a lot of the modern or the new techniques have been pioneered by amateur people because they want to get away from needing a lot of laboratory equipment. And so it's a place that anybody can jump in and get involved. There are several reasons to want to be working with fungus. I mentioned the soil building. Recycling would be another one. Food is an obvious one. Fungus is a very good source of protein, and there's some very unique amino acids that they produce. It's also for medicine. We're familiar with probably the most obvious one would be penicillin, is part of the fungal kingdom. And that, but there are lots of fungus. The big ones that are popular would be like the reishi mushroom, cordyceps, lion's mane, turkey tail, all of these things. And largely, they're from an herbal standpoint, they're adaptogens, so the things that fortify the body and help the immune system. And so the nice thing about this is that you can grow these things yourself and use them yourself. And most herbalists will tell you that herbs that grow in your own neighborhood are much better, much more powerful for you than things that are imported from far away. So there's a lot of work that can be done in capturing native fungus that's growing in your neighborhood and bringing it in and working with that. There's no reason necessarily to go to the herb store and buy something from China or far-flung anywhere. There's plenty of stuff right in the neighborhood that's going to be useful, and it can be eaten. You can make tinctures. You can do a lot of different things with it, and you don't necessarily have to produce the fruit body. Let me grab a couple things here. So this is a big portobello cap. Most of you are familiar with these. We can look at this in a minute. I want to get down off the stage here. There's not a tremendous amount of people, but in here you'll see the gills on the underside. Now this is where the spores are produced. So just briefly, the life cycle. So I just got a weather alert, so they may be closing things here shortly, but I'll be here anyway, so we can talk. But if you think of this as a spore, okay, that's the way fungus reproduces. So it's a life cycle, so you can jump in anywhere on the circle to understand what goes on. But in general, what we are used to thinking of as a mushroom would be something like this, which is the fruit body, but it starts with a spore like this, and the spore is a pretty perfect storage unit for the genetic information. And the spores can be they have activated spores from the pyramids, right? From any, they can last a long time. This mushroom produces millions and millions and millions of spores. This particular one, this actual piece, right, is going to make millions of spores. And any one of them can germinate. And when it germinates, it'll turn into a long filament, a hypha, a mycelium, a cell, and then it will keep dividing. And the thing with mushrooms is that they digest the world from the outside, right? So they don't take, let me put it this way, the mushroom will have the spore, when it germinates, it has a lot of genetic potential, and it can read the environment, it can understand what elements are present in the soil, for example, or in the tree, and it can produce any number of combinations of enzymes, primarily is what it does, and then it excretes these enzymes outside of itself. They break down the soil, they break down whatever happens to be there, and then those elements are reabsorbed into the fungus, but they're also made available to plants. So plants and fungus grow very closely together. Mycorrhizal is the term for that. Almost every plant has some mycorrhizal fungus associated with it, and most plants, it's essential that they have this for them to live. Now plants photosynthesize, so they take the sunlight, they intake carbon dioxide, and they give off oxygen. And they also, about 20% of they produce sugars in the plant, and at night, generally speaking, the plant will inject 20% of those sugars that it produces, it shoots back into the soil, and that is used to feed the fungus. And the fungus then dissolves, pre-digests material from the soil. And we now understand better the importance of the microbial environment for plant growth. Modern agricultural techniques generally just dump on nitrogen, phosphorus, and potassium, and all the elements are taken up and leave. When they harvest the crops, they leave the soil, and so it's depleted. And we know that the nutritional value of the crops has dropped dramatically over the years. It's pretty well documented. So better farming techniques is to care for the microbes in the soil, and they will produce what the plants need. So that lowers your input costs. You don't need all those fertilizers if the soil is healthy, but that's a costly process. And there's reasons why that doesn't happen as much as it should, but we haven't really understood that until lately. So that's another one of the very important functions. A lot of people who get interested in fungus, they want to encourage the health of the soil. So that's a kind of a different aspect. They're not particularly interested in getting something to eat, but they want to make the soil better. They can also be used to clean up toxins. There are strains, and the fungus can be trained to digest particular things. So you can take fungus into the laboratory, and you can expose it to the particular toxin that you want to break down, and the fungus will figure it out. And so it can be used to clean up oil spills. There's a guy, actually the guy that wrote this book, did an interesting project where he trained a strain of fungus to digest cigarette filters, which is a big problem. You don't think about it, but these things, they don't decompose in a normal time level. So that's all pretty useful. Yeah, so food, medicine, building materials, people are making fungus. There's some interesting work being done making some substitutes for leather, for example, out of fungus. It can be used to make building blocks. It can be pressed. You'll see in some of these things when we look at them that some types of fungus are very resilient. This, for example, is a reishi mushroom. This is several years old. Most of the time you think of fungus as like that mushroom I had, which you're used to, where they come up in your yard and they're around for a day or two and they dissolve and they disappear. But these woody ones, they stick around for years. And these are the cell walls of fungus are made of chitin, which is a starch and is one of the hardest naturally occurring substances. So this can be grown, the mycelium can be grown into sheets, can be used as insulation, can be used for a number of things. So there's a lot of research being done in that aspect also. Let's see, what else? Yeah, bioremediation, I mentioned that a little bit. So areas that are contaminated, where the soil is contaminated, the fungus could be introduced. Normally you wouldn't eat that fungus because the contaminants are going to be absorbed into the fungus, but it's a way of cleaning the soil, and then you have to get rid of and dispose of those fungus. But in the process, they're going to break that material down and make it available to other organisms and return it to the earth more quickly. But this is a very good medicinal mushroom. This is a little, when you look at this is a little zine that the guys from Radical Mycology developed this. And if you go to archive.org and type in Radical Mycology, it'll come up. There's a copy there that you can download of this. And it's a very good introductory thing, so you can have it in print. a lot of the information that is going to be useful to you as you start to actually want to grow these things. And then the final thing, well, probably not the final thing, but that I'm interested in also is using fungus in creating art of various kinds. And we'll get a closer look at this later, but this is the kind of crazy stuff that I like to do with them. So I grew this. This is grown actually on a pile of junk mail that I boiled and then inoculated with reishi mushroom again. And then as this was growing, I stuck some crystals and little man in there and stuff. So this will eventually turn into something a little more pretty than that, but it's pretty interesting as it is. Now this is probably one of the easiest little projects to do, and again, you can get a closer look at this in a minute. There actually is a nice little bouquet of oyster mushrooms coming up in here, and this is grown on toilet paper. So basically, all you do is get some cheap toilet paper, dunk it in boiling water, set it aside until it cools, and in that tube in the middle, you dump some grain that's been inoculated with the mushroom, wrap it up in a plastic bag like this, seal it with some polyfill, which I'll talk about in a minute. But anyway, you make a little filter on this so that it can get some air. The thing is, mushrooms are not plants. Okay, plants, like I said before, they take in carbon dioxide and they give off oxygen. Animals, like us, we do the other thing. We take in oxygen and give off carbon dioxide, and mushrooms do that as well. So they're much more closer to being animals than they are plants, but they're their own thing. But they're not plants. But in here, and you'll be able to see what, some examples when we look a little closer at this. When it grows in the bag and it doesn't have enough oxygen, it just grows in kind of a weird amorphous blob. And then once you open it up and give it some more fresh air, then it'll actually form the mushroom. And the mushroom then produces spores. Most of the mushroom life cycle, once this little spore germinates and it turns into it's called a hypha, so it's a single filament, and that has, and generally speaking, there's a lot of variation, but that hypha will have like half the genetic complement. Okay, so it's like sperm and an egg, it has half of what it needs. So that mycelium will have to kind of reach out in the neighborhood and come in contact with another spore that's germinated, and those two will get together. They'll exchange genetics, and then they can become have the full complement of the genetics. And it grows most of their life, it grows in this little fibrous network in the soil primarily. And then when the conditions are right, and it varies from species to species, but generally when they're running out of food, and then when the temperature and the humidity is right, that those fibers will weave together and they'll form that mushroom that we're familiar with, the stalk with a cap on it. But that's the same thing. That's all little mycelium, but it's woven together into this shape so that it can get up off the ground. It can produce the gills under the mushroom. The spores will form on the gills. The spores drop, they blow, they land someplace. If it lands in a good place, it'll germinate. It'll turn into mycelium. The mycelium will grow, and then it does it all over again. So when you're cultivating them at home, it's just a matter of getting a pure culture to start with, so you have only cells of the mushroom you're interested in, and then you give it the kind of food that it wants, you give it the temperature it wants, and you give it the airflow that you want, and the mycelium will reproduce. And then generally you change the temperature a little bit, cool it down a little bit, give it a lot of fresh air, and that will cause it to say, hey, maybe it's time for me to fruit, because I want to produce more spores, and they'll go on and propagate themselves. So in order to do that, there's a lot of competition. There's right now, probably every breath you take, you're inhaling hundreds and thousands of spores over the day. It doesn't generally cause any problems because we've been on Earth a long time, we've learned to deal with it, and it's fine. But there's a lot of competition. So there's all these spores floating around, and they're all looking for a place to grow. So when you want to cultivate it, the basic process is you get a pure culture, and then you propagate that culture, and then you introduce it generally first into grain of some kind. It can be any grain, it could be rice or wheat or anything. And then once it grows through that, the grain, you can use that grain to inoculate more grain, or you can use that grain to inoculate sawdust if the mushroom happens to be one that will digest wood. So those are the most common things it grows on commercially probably is either straw or wood chips. The common white button mushroom is kind of a manure lover, so it grows generally in composted sawdust, and of horse manure is what they grow that on. But that's really, there's not much point in growing white button mushrooms when you can go to the store and buy them for $1 a pound. But you can't generally go to the store regularly and get, say, oyster mushrooms even, because they don't ship well. They're very delicate, and so it's a lot easier to grow them yourself. But the oyster mushroom is also very rapid and a very strong grower. So when you introduce this culture into a growth medium called a substrate, so maybe sawdust, then it's going to take off and grow through that. And you want to have it. You want to give it a head start against other competitors. So typically, you would want to pasteurize those substrates, pasteurize the sawdust. And pasteurizing just means you're going to heat it up to like 160 degrees for an hour or so, and that's just going to knock back a lot of the already existing spores and things in there, and to give you a head start when you put your material in there. If you want to use some other medium or something that has more food in it, that's going to be more hospitable to all kinds of things, then you want to sterilize it, and that means you have to put it in a pressure cooker and kill off everything, and then just introduce what you want to introduce. So that's the basic system. This would be another example of just the way you could do it. So this is just a plastic jug. I cut it in half and then taped it back together, but it's just polyfill on the top, so that's just going to let air come in, but keep the other competitor spores out. So that's basically the process, whether it's in a jug like this, or in a bag, or in a liquid culture. Now that liquid culture is the big thing that changed the face of home cultivation, really, because in the past, in order to do this work, you had to build a sterile little environment, which mainly meant getting a real high efficiency air filter, blowing air through there, so you basically have a sterile air flow in front of this filter, and then you can pour agar into petri dishes, and that's just another medium that your material is going to grow on. And so that was kind of a little costly to do that, and but so it's people that were looking for cheaper and easier ways to do it came up with the liquid inoculation technique, and I'll explain that to you briefly here. So a real basic way that you would do this kind of thing is you get a pure culture, and so this is a petri dish that has mycelium growing on it. This is I don't know what this is. I got a lot of you wind up a lot of this stuff around your house with this. But anyway. So you scrape in a sterile environment, which could be in front of an airflow hood, or this big box that I have here is actually a work area. It's basically a glove box. So it's just a big plastic box, and I put those holes inside. So then when I want to work in there, I clean that out with alcohol, put the lid on, put rubber gloves on, reach through those holes, and do the work. And now you can also do it, if you're careful, you can do it in a real still room. There's ways you can do it that are very low tech or no tech ways, but it just depends what you want to do. And some things are easier than others. But the basic process would be to take a petri dish like this, which you can buy this stuff online from people that produce it, or I can give it to you or whatever. And you basically take a hypodermic syringe that has sterilized water in it. You open this up, you squirt a few drops of water on there, scrape around on it, suck it up into the syringe, and you've got cells in there that you want. Okay. Then you take that and you inject it into a liquid like this, which is basically a mild sugar water solution. It can be honey, it can be corn syrup, it can be a lot of different things. And then it grows in here. And all you got to do to keep this going is swirl it around once a day or so and keep giving it some oxygen so it can keep growing. Then you can suck, you can prepare a grain jar like this one. It's got popcorn in it, which you can use. I like popcorn because it's tiny, doesn't get all over the kitchen, they're pretty big. But you might want to use millet, for example, which could be tiny, a little more difficult to work with, but you get lots more inoculation points when you go to transfer it to another substrate. But anyway, then if you take the syringe again, and you can do all of this in the open air. The big advance was the development of this lid, which again was just an amateur guy and basically what this is, a regular canning jar lid, and you drill 2 holes in it. One hole you cover with a couple of blobs of RTV silicone, which just means it's high temperature silicone, room temperature vulcanizing. So you can get that in any hardware store, RTV silicone. So you put that on one hole. The other hole you stuff polyfill in. Polyfill, you buy a big bag of it for $2 or $3 at the fabric store and use it to fill pillows or whatever. So then you have this sugar water solution with the mycelium growing in it. You've got a grain jar like this that was pressure cooked, so it's sterilized. Then you can take that syringe, fill it up, and squirt a milligram or so of inoculant in here, and then in a week or 2 it'll grow through this grain, and then you can use this to inoculate something else. Or if it's a medicinal mushroom, you might stop there because you've got the stuff you want, and then you can just dry it out, grind it up, cook with it, make tea with it, or you can make a tincture. You can do all those things, but you don't have to go through the process. In order to actually get a fruit body, then you have to have another sort of a chamber where you can control the temperature a little bit, you can control the humidity, you can maybe keep the air clean. So all those things is just stepping up a little bit, in complexity, but it's very easy. Like, I don't know what they charge for Reese mushrooms in the store, but I don't know, they're probably $20 or $30 a pound, I don't know, maybe something like that. But you could grow unlimited quantity of that mycelium on your preferred grain, if you like rice or rye or wheat or whatever, and then just eat it that way and get the benefits from it. Yeah, I guess maybe if you just want to come up and look at some of this stuff, I can get off the microphone. We don't really have to keep doing this, and take a look at what these things are. I don't know what your interest is in this. If you have if you want me to go into more detail about one of these aspects of medicinal use or home, artistic use, building materials or whatever. Yes, John. So that's a big chunk of reishi grown on sawdust last year sometime. But it's pretty rugged stuff, and as you see, it you can see the potential there for building material. Yeah.
Could you do everything you need to do in a day to produce your own amount of medicinal or edible mushrooms?
Yeah, I think so. I mean, like I say, the tricky part is getting the pure culture to begin with, which you can grow. I mean, you can buy this kind of stuff. Like you can buy a petri dish of some particular culture, or you can buy. A lot of guys will supply you with a syringe with a liquid inoculant in it, and you can use that. But that costs you maybe $20. So if you wanted to grow reishi, okay, you go online, you buy a reishi syringe from somebody for $20, and then all you have to do is bring that home, prepare some jars of that sugar water, squirt it in there, wait for a week or so, and then that's going to have it's going to reproduce in that liquid. And then you can just start sucking that up and shooting it into grain, and off you go. So it's all pretty. It's pretty simple. Yeah, after a while, mostly, I mean, I like to use like just corn syrup because light corn syrup because it's clear. So I know I have a clear liquid. After a while, and I think maybe in some of this stuff, after it grows in there for a while, it will start to change color, usually yellow up, because the mushroom is producing enzymes and waste products and stuff that is going to change the color. Still going to be good, and a lot of people want that. There's a lot of medicinal uses for those other byproducts, the waste products that the mushrooms produce. And there's even been some interesting work done in producing medicines for you. So, for example, it'd be possible if you had a cold or some kind of infection, say some kind of sinus thing going on, you could introduce some of that material from your nose, put that on a petri dish along with the mushroom. The mushroom's going to grow over there, it's going to encounter that, and then it's going to say, whoa, wait, what am I going to do about this? I don't like it. It's going to figure out how to overtake it and either encapsulate it or detoxify it. So some people have worked with that to make a medicine for themselves. So then that particular strain of fungus that you produced is going to be adapted for whatever that organism is that you're trying to fight. So there's a lot of promise there, particularly in, as we're facing difficulties with the antibiotics that we already have, that they can figure out a way to take care of that. Yeah.
Oh. What about want to start mushroom without buying the inoculant?
Oh, okay. Yeah, actually, that's a good point. There's a couple of ways you can do that. Like that mushroom cap that I showed you before, for example. If I wanted to, portobellos wouldn't be my first choice, but say with an oyster mushroom, you can. The inside of the mushroom is going to be basically sterile, right? It's going to have just the mycelium is going to be in there. So you can take, again, one of these syringes that you use a large gauge needle, 16 or 18 gauge needle. You can take the stem of the mushroom, clean it off with alcohol, jam that needle through there, and then when you pull it out, it's going to have a little plug of mycelium stuck in the needle. Then you just take that, shoot it into the sugar water, wait a little while, and you've got your culture. So the other thing that surprisingly, dried mushrooms, even that rock hard black reishi I showed you, potentially, it's still alive, it's dormant, really. You can take a cutting from inside of that, or a chunk of that, soak it again in water, say maybe first to get it hydrated, then take that, soak it in hydrogen peroxide for a little while, because hydrogen peroxide will destroy spores, but it won't destroy mycelium. There's too much mass there, so you can take that chunk of rehydrated dried mushroom and put that on a petri dish and wait a while, and it will eventually come back to life and start to grow again. And so you can do it that way. So you can capture your own, and that's really One of the things, actually, one of the bags here I have is an oyster mushroom that I collected last fall down in Meigs County, and a friend of mine, and I did exactly that. So captured that particular one, and so grow that out and see how it works. And obviously, things that grow in your neighborhood or are adapted to your climate are going to do better. There's a whole other aspect of this, just like growing them outside, growing them outdoors, growing them on logs, and so you don't have to build a grow room that's temperature and humidity controlled and all that. You can just inoculate logs and have them out in the yard, or they do this with shiitakes, are done that way all the time. People cut logs and then use this sawdust inoculant and drill holes in the log, stuff them full of that stuff, and then let them sit for a couple of years, and they grow through, produce the mushrooms, and they grow that way. But the thing with that is that it's only going to produce mushrooms when the conditions are right, and that varies in the area and with the strain. If you want to have mushrooms all the time, then you have to create an environment that you can control.
So we get about 10 minutes left. Just a little bit less than that for questions.
All right.
I have one actually. I might have missed it, but are you able to grow morels?
Morels can be grown. It's not really worth the trouble.
Oh.
And generally, the thing, morels have an extra step in their life cycle. Now, when I mentioned to you the typical cycle where you have a spore, it germinates, it crosses with another spore, it grows, and the conditions are right, it produces the mushroom and makes more spores. Well, the morel does a little different thing. It produces the mycelium, but then it forms a body called a sclerotia, which is like a little stone. It's like a little pea-sized knot of material. And these then lie in the ground for years until the microclimate's right in that little area, and then it'll produce a fruit body. So that's why, when you go hunting morels, you don't always find them in the same place. They're there, they're all over the place. They're in the soil. These little beans are under there waiting, but they're only going to kick into the reproductive phase when the conditions are right. There are some people growing, there is an outfit or two that grow morels indoors, but they're they don't get a very good yield from it. And they're not very well understood. Genetics, we have a tough enough time thinking about genetics with two sexes. Mushrooms have you can have 20 or 30 different sexes. So it gets pretty complicated. And actually, like with morels, there's black morels, yellow morels, and white morels. But some researchers have produced, under conditions, careful conditions, produced sclerotia from one particular mushroom, planted them outdoors, and then all these different kinds of morels come up, black ones, white ones, yellow ones, and nobody knows why they decide to express. So I think, morels are pretty much better just left as a mysterious thing that happens in the spring, and you just take, enjoy them for what they are.
All right, so we are going to have to wrap this up, if that's all right. So, do you have anything left?
No, like I say, I'm here, so we can. I don't know what's at in the next, but if you want to come up and look at some of this stuff, we can haul it off to the side there. But again, look up radical mycology. Remember that word. Look at the archive.org, and then you can find us on Facebook, or if you want to give me your name and email here, I'll get in touch with you, and happy to do that.
And we are going to be shutting down.
Okay, well, the show there is over. That talk was filmed today at ComFest in Columbus, Ohio, and we got rained out. When it rains a lot, as it did today, they closed the park where we were having the festival because they were afraid of damage to the grass and so forth from traffic and the wet. So anyway, I'm obviously not there at the festival anymore, but I thought I'd add on a little bit here and give you some close-up of some of the material that's in the that I showed at the talk that you really couldn't see. So I'm just going to briefly go through my little presentation box of show and tell items. So here we go. Okay, this is the glove box we looked at a little bit. You can see these holes here. There's one and the other one. Actually, the way that I cut those, which I think is kind of interesting, a trick I learned from another mushroom cultivator, William Padilla, Padilla Brown, I think. Anyway, basically you take a coffee can and heat that up on the stove. You want to get it nice and hot, and then you can just poke it onto put it on the plastic, and it burns right through. So that's the glove box. We'll open her up and see some of the other items that we looked at. This is the oyster mushroom. Let's see if we get a little better shot of the actual. There we go. There we go. So you can see the. This is golden oyster mushroom, happily growing on toilet paper. Here's my little in progress sculpture. This is a reishi mushroom growing on boiled junk mail. Did this last fall. Here's a little closer look what's going on there. So that's that. Here's a bag of sawdust spawn. This is sawdust that has oyster mushroom growing through it. This is a variety called Phoenix, and you can sort of see maybe the little knots of mycelium that are forming there, and that's the beginning of the mushrooms wanting to fruit. But because they're in this, still in the plastic bag, they're not going to form the nice pretty mushrooms until poke some holes in this, and then the mushrooms will sense the atmosphere, and they'll come out looking for more oxygen, and they will grow into mushrooms and blossom out the side, more or less. This is a close-up of the bag that this is the oyster mushroom that I captured from my friend's forest there down in Meigs County. And this bag is kind of a homemade affair with a little polyfill twisted in the top. So this is the initial stage where you get it growing in a kind of an oxygen poor atmosphere. Mycelium is happy enough in there until it needs to fruit, and it needs more air. So we'll give some more of that later. This is the block of reishi mycelium from a year ago, but just to demonstrate that all the mushrooms are not mushy and soft. Some of them are hard and woody, and this stuff is, besides being a good medicinal, could potentially be a building material. This is a petri dish with some mycelium growing on it. This little mark here is where I scraped up some mycelium to make liquid culture at some point. That's that. This is a commercial mushroom filter bag. This kind of plastic will withstand pressure cooking and sterilizing, autoclaving, which the other bags will not. They will withstand pasteurizing temperature, however. So they're cheap. These bags are expensive. Okay. Then this is a jar of dried grain spawn. So this is a medicinal mushroom, red belted polypore. I have not gotten those to fruit yet. I haven't really tried too hard, but basically the medicinal qualities are still present in this dried grain mixture. So this could be used as is, ground up and added to food, or make tea, or make a tincture. Here's a liquid culture, close up of that. This is let's see, this is a honey liquid culture, so it has a little more color to it than the clear white. This is a cordyceps culture growing in there, and the what's called the airport lid, which is really the latest improvement in the technology that allows you to do it out in the open air without having to worry too much. Here's the injection port. You squirt the stuff in through there. This is the air filter, again just with the polyfill stuffed in the hole. So that's good to go. Here's a close-up of the cooked popcorn. This would be again the airport lid, so the liquid from the other jar could be squirted in here, and this would grow through and be ready for further steps. This is another liquid culture, and there's a couple of marbles in there. I like to throw a couple of marbles in sometimes, so they, when I shake this around to aerate it, it will move things around a little more when you got those marbles to rattle around in there. The tin foil over the top is still on there from when it was pressure cooked, because when you want to keep that polyfill from getting wet, because if that gets wet, that's going to be an avenue for unwanted spores to get in and possibly contaminate your liquid culture. This is a close-up of that little jar of another little experiment with a plastic jug, so making a little environment, a terrarium kind of, so we'll get some mushrooms in there in a few days, I would imagine. Some of these things we didn't look at too closely in the talk, but this is just some other random petri dishes to show you some of the these are kind of old and just things that were laying around I brought to look at. Here's another one. This I think is red belt, and you can see this actually turns the agar. It gets this black or dark background. So the myceliums can vary quite a bit, but you can certainly see the fibrous network. Now this is how the mushroom grows. Spends most of its time in this condition, and only decides to fruit when it's time to go into reproductive phase. So this is basically the vegetative state. Here's another petri dish, and again you can see this has been sitting around so long that it's trying to fruit, it wants to fruit, and these little knots that are forming, that would be the beginning of the process, but they're not getting enough oxygen in here at the time to do it. This petri dish is sealed with a material called parafilm, which is a plastic waxy material that does allow some air to get through, but it keeps unwanted stuff from getting up under the petri dish, under the lid of the petri dish. Okay, it's another random mycelium. Here's yet another one. I think this is red belt also, and you can see these black little areas start to form as it matures. That's part of the process. Looking for this red belt actually is one that I also captured from a friend's property, and so this was done the same way, taking a few cells from inside of the mushroom and putting them onto the petri dish and then growing out. Let's see. Here's another little bag of golden oyster mushrooms. We'll see inside there. They're starting to become recognizable as what we would consider to be the actual mushroom fruit body. So over the next couple of days, this will I take this out of the bag and set it up so it has a little more room to breathe. It will produce bigger clusters. Eventually it will digest all that newspaper, or the toilet paper, and I'll get some good eating mushrooms. This is a coffee can I talked about before that I used to heat up and basically quickly burn a hole through the side of the plastic. I've done things like this before, trying to cut holes with a saber saw or hole saw, and that works, but you wind up cracking the plastic often, or you get a rough edge on it when you're done, and this eliminates any need to smooth that edge down. It's a really slick little technique. I'm glad I learned that. This is the portobello caps that I had just as an example of your typical sort of mushroom that we think of. So these are, I'm sure you've seen at the grocery store, delicious for eating. And these are the gills on the underside. You can sort of all mashed up, but this is with a spore-bearing surface. But this interior part here, if I split this open, that's going to be sterile. So if a person wanted to, you could capture this culture and use it, propagate it, and grow more of them. Here's another reishi that I grew. Reishis are fun because they're nice for me. I like to do the sculptural kind of stuff. So this one is many years old and still never treated with anything, so it still would be a good medicine. But with the reishi, when you control the carbon dioxide level in the grow area, when there's the way these things grow in nature, typically they grow on trees, so they would maybe start inside of a little cavity on the side of a tree, and then its first concern is to get out to where it can have enough space to spread some spores around. So in the beginning it grows in this what's called an antler form, and kind of looks like coral, and then you can see here in the early part there's no white part, which is the spore bearing surface. So this was in nature, this would say be inside of the tree and would grow out, trying to reach outside of the hole, and once it gets out, then I gave it some more oxygen, and then it would form this white surface underneath, which is similar in function to the gills on the other mushroom. These are little pores. This is an example of a mushroom that's called a polypore. So it has little tubes on the underside, and the spores are produced in there. So the mushrooms have many techniques that they have developed over the eons to do their thing, which is basically keep growing, keep producing more mushrooms, keep spreading out across the earth. So they're hard-working little guys. Let's open this up. This is the kind of the key to the modern, more simpler way to do it. This is called an airport lid. So this is just a close-up regular canning lid that has a couple of holes drilled in it, and one is plugged with high temperature room temperature vulcanizing RTV silicone, and then another little hole that has some of this polyfill stuffed in it. So this provides some room for gas exchange. The CO2 that the mushrooms are producing can get out, and the oxygen can get in, but this will effectively keep out competitor spores and stuff, so the mushrooms won't have to fight off anything else. And this is remains of a bag of polyfill, so very inexpensive. Bag like big bag of this stuff is a couple of bucks at a fabric store or Walmart or someplace. Very handy. And then this is just a stack of petri dishes, as an example. So that pretty much covers the content of the mystery box, and hopefully that will be of some use to you to see that. And let's see. Turn this around here. Okay. So that's it for a little close-up of the material that I was showing at the presentation that we just filmed earlier today. So hope that's of some use, and I look forward to sharing more information with you. Okay. See you later.